Yeah, welcome to Vesta, where biology's the scene, breaking down cells, DNA in the machine. From Nucleic systems to how life began, follow the chain, understand the plan.
G'day everyone, my name's James Besta and welcome back to Vesta Biology. In today's episode we're talking about nucleic acids, the blueprint of life. We'll explore DNA and RNA, how they store and transmit information, and how this information finally gets turned into proteins. If you missed our previous episode on proteins, I'd recommend checking it out because today's content links directly to how proteins are made. Now let's get started. Nucleic acids, specifically DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are essential molecules that store, transmit, and express genetic instructions. Here we see the DNA on the left and the RNA on the right. DNA acts as the long-term genetic blueprint, while RNA is the messenger and worker that helps translate these instructions into proteins. Remember when we talked about how proteins are essential for cell function and structure? This process all starts here with DNA and RNA. DNA is found in eukaryotic cells inside the nucleus, packaged into chromosomes. Eukaryotic cells being what we generally find in multicellular organisms such as us, as our cells need to be much more organised. These chromosomes are made of DNA and are wrapped around proteins called histones, which are the ball-like structures in the bottom right picture. This is a good example of how proteins play a key role in our biological processes. DNA is a polymer made up of nucleotides, which are its monomers. If this sounds familiar, it's because in our proteins episode, we learn about amino acids being the monomers for proteins, just like nucleotides are for the building blocks of DNA and RNA. We've got two pictures here, a more simplified one on the left and the one on the right, which is a little bit more detailed. The main thing we need to know about our nucleotides are the three key parts. We have a phosphate group and we have the deoxyribose sugar if it's DNA or a ribo sugar if it's RNA. And then we have our four nitrogenous bases. We have adenine, thymine, cytosine, or guanine. It's important we get used to using their proper names and not just the abbreviated letters, as our bases we'll see again in some other chapters. For example, mRNA, it's going to use these bases to stabilize its structure before heading to the ribosome and then when it gets fed through the structure. So we're going to see these names pop up again. Just like amino acids in proteins are linked by peptide bonds, nucleotides in DNA are connected by phosphodiester bonds, forming the sugar phosphate backbone. This bond forms through a condensation reaction, something we also saw when polypeptide chains form. This process releases a water molecule as the phosphate group of one nucleotide forms a bond. This creates a chain with distinct directionality. We see in the diagram the strand on the left has the 5' end at the top. This means that the phosphate group attaches to the 5th carbon of the sugar, which you can count to. The 3' end is at the bottom, where the next phosphate attaches to the 3rd carbon. DNA strands run anti-parallel. One strand goes from 5' to 3' and the other runs 3' to 5'. We can see in the diagram that the pentose sugars are flipped, therefore the positions of the 3' to 5' change. This has been simplified but hopefully creates understanding where these terms come from. This arrangement is crucial for DNA replication and transcription, which we'll cover later. DNA strands form a double helix, held together by hydrogen bonds between complementary bases. Adenine pairs with thymine with 2 hydrogen bonds, cytosine pairs with guanine with 3 hydrogen bonds. We can see the different base pairings up across the center of the DNA. Note the lines denoting the hydrogen bonds. This complementary base pairing ensures accurate replication of DNA. Now these bonds aren't very strong, which means that when DNA wants to unwind, this is where they can do it. RNA, another type of nucleic acid, is involved in expressing genetic information. RNA differs from DNA in several ways. Firstly, it's single-stranded. Second, it contains a ribose sugar instead of a deoxyribose. And third, it uses uracil in place of thymine. This substitution occurs because uracil is simpler and allows RNA to be more reactive, perfect for its temporary role in protein synthesis. There are 3 types of RNA. We have mRNA, which is short for messenger RNA. It carries instructions from DNA to the ribosome. Second, we have rRNA, which is ribosomal RNA. It catalyzes peptide bond formation at the ribosome. Third, we have tRNA, transfer RNA. This delivers amino acids during translation. Here's a quick comparison between DNA and RNA. For both, they're going to use a monomer of a nucleotide. Strandedness, DNA is double, where RNA is single. Nitrogenous bases, DNA has adenine, thymine, cytosine and guanine, where RNA replaces the thymine with uracil. For a sugar, DNA has a deoxyribose, which means it's missing an oxygen off its carbon ring, where RNA is just a ribose sugar. For the lifetime, DNA is very long-lived, years to the lifetime of the cell, where RNA can be very short-lived. We're talking hours potentially to a couple of weeks. The function of both is very different. DNA is going to store genetic information, where RNA is going to express this genetic information. Now let's connect everything with the key principles of molecular biology. It explains how genetic information flows from DNA to RNA to a protein. First, we have transcription, where in the nucleus, RNA polymerase reads the DNA template to create mRNA. Second, we have translation, where in the cytoplasm, the mRNA binds to a ribosome, tRNA delivers amino acids to build the polypeptide chain. This is going to fold into a functional protein, just like we discussed in the proteins episode. Stay tuned, we'll dive into transcription and translation in a future episode to give these processes the details that they deserve. Here's an exam question for you to have a go at. Describe the structure of a DNA nucleotide and explain how nucleotides link together to form a strand of DNA. 3 marks. This question tests your understanding of the components of a nucleotide, phosphate group, deoxyribose sugar, nitrogenous base, and how they bond to form a DNA strand. It's a question you might encounter in VCE exams. First, I'm going to highlight the task words and content words. I've discussed doing this in a previous video on answering short answer questions. Pause it to give yourself a bit of time, and on the next slide, I'm going to go through an example response. So here's an example response to that question. A nucleotide consists of 3 components: a phosphate group, a deoxyribose sugar, and a nitrogenous base, adenine, thymine, cytosine, or guanine making up those bases. That's going to get you your first mark. Second, nucleotides link together through a condensation reaction which releases a water molecule. Second mark. This reaction forms a phosphodiester bond between the phosphate group of one nucleotide and the sugar of another. This is going to create the sugar-phosphate backbone of the DNA strand. Third mark. Let's quickly recap what we've learned today. First, DNA and RNA are polymers of nucleotides, just like proteins are polymers of amino acids. Vice versa, we've got the word monomer, which means the nucleotide is a monomer of DNA and RNA. Nucleotides join together through phosphodiester bonds, releasing water through a condensation reaction. One strand of DNA helix is in the 5' to 3' direction, and the other is anti-parallel, therefore it's running 3' to 5'. DNA is double-stranded, RNA is single-stranded. RNA also uses uracil instead of thymine. DNA is long-lived while RNA is short-lived to suit their different roles. The central dogma describes how genetic information flows from DNA to RNA to a protein. Well, that's it for today's episode. If you found this video helpful, be sure to like and subscribe for more Best of Biology content. If you've got questions, drop them in the comments below. I'd love to hear biology questions or lesson ideas from you guys. Thanks for watching, and I'll see you in the next episode. Peace out.
Best of biology, when how does unite? A journey of discovery, reaching new heights. Every molecule, every cell we explore, unlock the universe, see what's in store.